Imagine waking up in a world where humans no longer fear disease, where babies are born immune to cancer, where memory, strength, even intelligence are editable like files on a computer. Sounds like science fiction? It's not. It's the very real, very present revolution of genetic engineering. And unless we understand what's coming, we might sleepwalk into the biggest change to life on Earth since evolution began. Let's dive into the code behind every living thing and discover the tool that could either save us or rewrite what it means to be human. We've been hacking biology for thousands of years, long before we had any idea how biology actually worked. From the first farmers who bred stronger wheat, to the dogs we turned from wolves into companions, to the cows, corn, and carrots that no longer resemble their wild ancestors. Through selective breeding, we changed nature to suit our needs. But it was slow, clumsy, like editing a novel by ripping out pages and hoping for the best. Then came the 1950s. The discovery of DNA, deoxyribonucleic acid, cracked life's source code. A spiral staircase made of 4 letters, A, T, C, G, arranged in endless combinations. Those letters tell cells how to build everything. Your eye color, your resistance to disease, your risk of Alzheimer's. It's all encoded in 3 billion base pairs wrapped in every cell of your body. And once we could read it, we started wondering, could we write it too? By the 1970s, scientists began inserting genes from one organism into another. Glow-in-the-dark jellyfish genes went into mice. Human insulin genes went into bacteria, creating lab-made insulin that would save millions of diabetic lives. By the 1990s, genetically modified crops were on supermarket shelves. By the 2000s, scientists could clone animals, like Dolly the sheep. But it was still slow, still expensive, still unpredictable. Then came CRISPR. CRISPR is a biological tool, but calling it a tool is like calling a rocket a paperweight. Discovered in bacteria, it was originally part of an ancient immune system, one that remembered past virus attacks by saving fragments of the invader's DNA. When that same virus attacked again, the bacterium would whip out a molecular weapon, a protein called Cas9. Cas9 would scan the DNA inside the cell, find the exact viral match, and slice it apart like scissors. When scientists realized this system could be programmed, when they understood you could guide Cas9 to any genetic sequence and cut it there, they unlocked one of the most powerful editing tools life had ever seen. The power of CRISPR lies in its precision. Think of older gene editing techniques as using a sledgehammer, effective but messy. CRISPR is more like using a scalpel guided by GPS. Scientists can now switch genes on or off, fix broken genetic code, or insert new instructions into any type of living cell, bacteria, plants, animals, even humans. One of the most promising uses of CRISPR is in medicine. For instance, in 2015, researchers successfully removed the HIV virus from infected human cells in the lab. Just a year later, CRISPR was used to eliminate more than half the virus from live rats by simply injecting it into their tails. A future where viral infections like HIV or even herpes can be erased at the genetic level is now a tangible possibility. But CRISPR doesn't just target viruses. It might help us defeat our oldest enemy, cancer. Cancer cells are notorious for hiding from our immune system while multiplying uncontrollably. With CRISPR, we can reprogram immune cells to be more aggressive, smarter, and better equipped to detect and destroy cancer. In the future, cancer treatment could mean getting a personalized shot, a few thousand of your own cells re-engineered to fight for you. The first human clinical trials for CRISPR-based cancer therapies began in 2016. Since then, the pace has only accelerated, with trials expanding to include diseases like sickle cell anemia, muscular dystrophy, and even heart conditions. And what about genetic diseases? Those inherited errors coded deep in our DNA? There are over 3,000 known single-gene disorders, many of which are deadly or cause lifelong suffering. CRISPR opens the door to correcting these errors at their source. Scientists are already designing tools that can change just a single letter in the genetic code. Fix that letter, and the disease is gone. This means future generations may never need to suffer from the same illnesses we've come to accept as part of life. But here's where the story gets complicated. Editing somatic cells, meaning cells in your body that don't get passed on, only affects the individual. But editing germline cells, like embryos or reproductive cells, means editing the blueprint of future generations. These changes are heritable. They rewrite the human story. And that future is no longer hypothetical. In 2015 and again in 2016, Chinese researchers edited the DNA of human embryos. While those embryos weren't brought to term, the experiments proved the technology works and exposed just how much we still don't know. Ethical lines are blurry. Should we use CRISPR to eliminate disease from the gene pool? Or is that a slippery slope toward designing babies with preferred traits? Height, intelligence, appearance? We're standing on the edge of a revolution that could redefine evolution itself. And once we cross it, there's no turning back. If this video sparked new thoughts in your mind, don't let them fade. Drop your biggest realization in the comments below. I read every one. Subscribe to Deep Synapse for more explorations into the future of science, the mind, and what it means to be human. And if you believe these ideas matter, share this video, because the future doesn't wait, and neither should our understanding of it.